So we've got sound, we've got equilibrium. The other special sense we're going to talk about here is sight. And then we're going to talk about, in the lab specifically, receptors of touch. You use eyes in order to detect light. Excuse me. These specific light cells are called photoreceptors because they receive light. Broadly, there are two types of photoreceptor cells. Rod cells, then they're going to detect amount of light, and cone cells that detect quality of light. So these rod cells will detect how much light is present. These cone cells will basically tell you what color things are. Photoreceptors, especially cone cells, have pigments in them. As we learned in Bio 101, pigments will absorb very specific wavelengths of light. What pigment did we talk about in Bio 101? Do you remember? Starts with a C. Yeah, chlorophyll. Chlorophyll absorbed light in the red and the blue spectrum, but not in the green spectrum. So we're going to use these cells in order to absorb different wavelengths of light. Now, different animals have different types of eyes. And I say eyes sort of with quotes around them because we wouldn't necessarily consider all of these eyes. We have ocelli, compound eyes, and the single lens eye. Ocelli, as you can see, bees have 2 big eyes and 3 ocelli. The ocelli are going to be really trying to figure out where light is. Is it dark or is it light? The eyes of a planarian, a flatworm, are going to be like ocelli. They're going to tell it, is it dark over here, is it light over here? And it allows them to head toward the dark. Insects, a lot of insects tend to prefer the dark. Heck, what are those things called? Cockroaches, right? You turn on a light, you see cockroaches scatter because they don't like light. They're going to not only have compound eyes for detecting more complex views of the world, they're going to have ocelli to say light and dark, head toward dark. Questions? We have our compound eye. Compound eyes have thousands and thousands of what are called ommatidia. These are individual lenses. So they capture light coming in from a very specific direction. Have you guys ever looked, okay, it's like looking through a toilet paper roll. All right, you look through a toilet paper roll and you can see one thing in front of you very, very, very well. But you don't see anything outside of that. Now, what these guys have is thousands of toilet paper rolls all over. So they can get information all about them. Now, they're not getting depth. They're not getting terribly much, their brains, as you can tell in the fly, which is not very complex, have to interpret that information. So it is likely what they can get from this information is a great sense of movement. They know when something is moving along those tubes. That's through those single lenses. They can't make out a lot of detail, but they know something's moving. Have you guys ever tried to smack a fly? It's hard to do. They always see you moving. You could be moving very slowly, and they still see that movement. Yes? I don't know that. I know that they probably don't have terribly many cone cells to interpret different colors of light. Interesting. We'll have to try the experiment. I'll try that experiment myself. Trying to chase a fly down with a red object. If that was the case, they should make like all fly swatters red. And then the single lens eye, which is what you have. And we'll go into a little bit more detail on. The single lens eye has an opening called a pupil. At the very, very top, it's a little hole leading directly into the eye. There's a lens immediately behind it that's opened and not opened and closed. It's basically squeezed and released using muscles. You've got a liquid in the back that's called a vitreous humor. And then along the very back of the eye, you have a whole lot of rod and cone cells for detecting both light and color. Right at the back of the eye, you'll see that there is the space for the optic nerve and for blood vessels to come in. This small area right here, this is called the, this is the optic disc, fovea. I forget what that, oh, that's your blind spot. It's your blind spot because there are no rod cells or cone cells there to accept light. During the lab, we're going to actually measure where your blind spot is. Do you guys ever, do you know what floaters are in your eye? You got these little like specks floating around? What that's usually caused by is the vitreous humor being a little bit denser in regions. Basically, it's holding on to itself and creating a little cloud. So that if you have this cloud of this thicker area of vitreous humor, it's not completely clear, it's a little opaque. As it passes by the by your cells, it's stopping light from getting in, and it's making it harder for them to interpret the world around you. Now, the closer they are to these cells, the smaller the impact is, because you're only blocking a few cells, and the rest of the cells compensate. You'll notice with these floaters that even though you may have one, you start to block it out after a while because your brain begins to process that information and say, based on all of the information I'm getting from all these other cells, here's what the world looks like. Adjusting that lens is what allows you to focus on an object near and far. In fact, we'll go into this more in the lab. If you were to look closely at something you were reading and then drop it down and look far away, it takes your eyes a minute to adjust to that difference. Right now, I can read that safety first perfectly, it looks great, and then I glance over to this and this is a little bit blurry. Suddenly this focuses and then I glance back and that was blurry for a second. Our eyes are quick to make that change because we're predators. That means that we are constantly using our binocular vision in order to figure out how far away something is and get details about different objects at different distances. Changing the shape of that lens does take a muscle though. So if you're constantly changing the shape of your lens to focus at different positions, you're going to get a headache just from muscle strain. Oh, I did not know that everybody can't make their eyes go blurry. Oh, interesting note. In humans, and mammals, we use muscles to control that contracting or expanding around the lens. Fish and invertebrates actually move the lens itself forward and backwards. If you ever look at a fish's eye, it's kind of weird because that lens is almost utterly detached. Yes? It could. Humans, you guys, and a minority of mammals can see in color. Most mammals cannot see in color. Fish, amphibians, birds, and reptiles can see in color. Typically, if you see a colorful animal, it's likely can see in color because it's using that as a mate choice. So it's a secondary sexual characteristic. But a lot of mammals you don't see with bright, flamboyant colors. Look at tigers. Tigers are striped. Why are tigers striped?
Look like grass.
Yeah, to look like grass. But dude, they're orange. It doesn't look much like grass. If, though, you looked at them from the perspective of their prey, they look exactly like the grass because the prey can't see that orange. All they see is, we think, just the patterns. So to see colors, we use these photopsins, the cone cells. There are 3 different pigments in the cone cells. We've got blues, greens, and reds. That gives you a wide assortment. Abnormal color vision is caused by mutations in the photopsins. Maybe the red photopsin doesn't work, at which point you only see green and blue. So all of these up here are basically, this is what you would see if you were normal-bodied. These are simulations of what you would see for different color blindness. So if you can't see anything in this one, don't worry. It's a simulation. It's to show you what it would look like if you were colorblind. So there we go. We've got two types of cells in the eyes, rods and cones. Rods are going to be used for detecting amounts of light, so they're going to be your low color vision. Your low light vision. Have you guys noticed that when it's dark, you're in a dark room, you don't see colors well, but you can see shapes? That's straight up due to your rod cells. Is it true what? That I don't know. Cone cells are going to be used for detecting light. There are three distinct kinds of eyes. You have a single lens eye, but you might have ocelli, which are used for detecting light. Well, you don't. Insects might have ocelli that are used for detecting light. And you could have compound eyes, which are used for detecting movement. Questions or concerns?